like in movies or people from New York, it's like it's at this street and the cross street of this. So knowing these sections or knowing these divisions can be really helpful in sort of making it that internal map in your brain or in your mind's eye of those little neighborhoods. And it's the same thing when we talk about the divisions within the brain. We're going to introduce you to the 5 neighborhoods of the brain, and we'll discuss them, in ascending order. I also want to mention or just remind that the higher structures have less reflective and more complex functions, and then the more recently evolved structures tend to be the opposite. So here's that bottom up piece. So let's just take a look here at this, and we'll start there at the bottom, spinal cord, and then into the hindbrain or that green area. So the hindbrain is comprised of both the myelin and the metencephalons. And inside here, you will find things like the reticular formation, and kind of study on a cellular level how it all works. But in general, there's synthesis of a neurotransmitter, there's storage of it, there's movement of it, which I didn't include in the action, release of it, that postsynaptic cell receiving it, and then once it's done doing its thing, the inactivation and what happens in the aftermath of that. Here's a bit easier picture, I think, to follow. So for a neurotransmitter to have an effect on a postsynaptic neuron, it has to bind successfully to its receptor that's embedded on that membrane of that postsynaptic or that receiving cell. So here we go. So we have drugs. So don't worry about this picture is showing mechanisms of drug effects, but I'm looking at the inside here to make this point. So we have neurotransmitters that are synthesized, that are then packaged into these vesicles, that then move down the sending cell and fuse and open up and release all that something like serotonin supply at the place of the synapse, or by stimulating a dopamine type 2 receptor directly, remember that one's inhibitory, they could reduce cravings for alcohol. So specifically, drugs that would be considered dopamine type 2 receptor agonists could help reduce alcohol intake in high alcohol preferring, in this way it was a rat model, whereas dopamine type 2 receptors antagonists increased alcohol drinking in this inbred animal strain model that they were using. And that was in the early 90s. So, we've come a long way since then. This has been substantiated, repeated, done on a higher scale. We know a lot more about this now. So what's the main neurotransmitter involved in the biology of reward? That would be dopamine. And so Blum et al. Availability, they seek contact, but they resist. There's a lot of anger and reactivity. This is the one that's associated with that type of pathology. It's not causal, but it is the one when we look back and we look at these various attachment patterns, we can kind of see, again, maybe more biologic vulnerability for the development of that if one has that kind of pattern. So in the past few decades, attachment theory, I think, has really evolved, and has become more modern. They provide updates to Bowlby's theory, and it's now really looked at as the neurobiologically informed attachment theory. This would be Shore et al. So it's still grounded in developmental, affective, and social neuroscience. The modern theory really shows or posits that it's the hardwiring of an infant's early developing brain, and even more specifically, their right brain, that's imprinted